Hello, my name is Cindy Atwell, and I'm the head of business development and alliance management at Precision BioSciences. We are a clinical stage biotechnology company leveraging a next generation genome editing platform for the development of human therapeutics. Precision BioSciences is a public company, and I will be making forward-looking statements today. Please refer to the disclosures and risk factors in our SEC filings. It has been an exciting couple of years at Precision, during which we have built a world-class team of 230 employees. We've established a GMP manufacturing facility to support our off-the-shelf cell therapy clinical trials, and importantly, have smoothly transitioned from a preclinical development to a clinical stage company, with soon to be a total of 4 allogeneic CAR T therapies in clinical studies. On the gene correction side of the business, we've entered into a transformational deal with Eli Lilly for development of in vivo gene correction therapies with DMD, named as the lead program. The underlying foundation of Precision BioSciences is our genome editing pioneers and proprietary genome editing platform we call ARCUS. ARCUS was invented at Precision and was developed with an eye towards clinical translation without unintended changes in the genome. We are focused on addressing serious diseases through unlocking the full potential of our ARCUS genome editing technology. We are seeking to apply ARCUS to cure rare diseases, making a permanent change to patients' genomic DNA. And we are seeking to overcome cancer through the development of allogeneic CAR T therapeutics. We chose to invest early in manufacturing, as we see this as a strategic advantage. And we have established GMP allogeneic cell therapy manufacturing as a core capability. In July 2019, we opened MCAT in RTP, a fully GMP compliant facility. We're able to produce all the key components across the portfolio for CAR T and in vivo gene correction therapeutics. And we are currently producing clinical trial material for our BCMA CAR T, as well as PBCAR19B programs. I'll now provide an overview of our 2 therapeutic business areas, starting with our off-the-shelf cell therapy programs. Our pipeline includes 3 first-generation CAR T pipeline products in the clinic, as well as a second-generation PBCAR19B program, which will enter the clinic in the first half of this year. Additionally, we are advancing 2 undisclosed hematological malignancy targets and 2 undisclosed solid tumor targets in our preclinical studies under our collaboration with Servier. In the first half of this year, we plan to initiate dosing of a combination study with our BCMA CAR T and GSI through a partnership with SpringWorks. We anticipate releasing interim data on our lead program, PBCAR0191, in mid-2021, as well as initial data releases on our CD20 BCMA programs later this year. Part of the reason that we're able to rapidly advance our cell therapy programs in the clinic is that there are a lot of similarities across the 3 therapeutics. Namely, that we use a proprietary one-step gene editing approach to insert the CAR into the TRAC locus. We simply switch out the gene inserted, which is the CAR targeting the antigens relevant to the type of tumor we are aiming to treat. This allows Precision to rapidly apply its learnings across our cell therapy pipeline, as well as to rapidly develop new off-the-shelf CAR T therapeutics. Our lead program is PBCAR0191, an allogeneic CD19 CAR T product. And we are currently testing this product for the treatment of relapsed refractory NHL and ALL. The primary objectives of this phase 1/2 study are to assess safety and maximum tolerated dose. Secondary objectives include clinical anti-tumor activity. And we're looking at the expansion, trafficking, and persistence of our allogeneic CAR T cells as well. Importantly, we are exploring approaches to drive the durability of clinical activity. This includes exploring opportunities to enhance allogeneic cell expansion, trafficking, and persistence, including enhanced lymphodepletion. In December, we released preliminary results from this study, including 4 patients from the enhanced lymphodepletion cohort. We saw a 95x increase in peak expansion with the enhanced lymphodepletion cohort versus standard lymphodepletion. With the enhanced lymphodepletion cohort, we saw a 100% overall response rate and a 75% CR rate at 28 days. Additionally, we saw a greater than 90% decrease in tumor size. The safety profile we have observed so far is encouraging. There's no evidence of graft versus host disease. As anticipated, we see infections associated with lymphodepletion conditioning and no grade 3 or higher CRS or neurotoxicity. We are rapidly leveraging learnings from our PBCAR-019 study and enrolling more patients into our enhanced lymphodepletion regimen. In parallel, we continue to evaluate higher cell doses, repeat dosing, and other novel methods to further optimize our dosing strategy. The mid-2021 PBCAR-0191 clinical update will include additional patients within the enhanced lymphodepletion cohort, as well as the clinical activity measures beyond 28 days. We are pursuing clinical development of our second generation CD19 product in parallel to the first generation. This is our CD19 stealth cell candidate, PBCAR19B, which includes immune-evasive technology and has shown to delay both T cell and NK cell mediated allogeneic rejection in vitro. We believe that delayed T cell and NK cell mediated allogeneic rejection is likely to result in improved persistence of allogeneic CAR T cells and potentially improved clinical activity. The IND was accepted by the FDA, and we expect to move PBCAR19B into the clinic in the first half of 2021. Let's now turn to Precision BioSciences in vivo gene correction pipeline, where we are utilizing ARCUS to make permanent genomic edits to patients' DNA in order to develop potential cures for serious rare diseases. We are collaborating with Eli Lilly to advance our DMD program, as well as 2 undisclosed targets. Additionally, we anticipate providing an update on our primary hyperoxaluria type 1 program in the mid-2021 timeframe. We have achieved in vivo large animal proof of concept with the other programs listed in this table, and ARCUS was invented at Precision really with a focus on clinical translation to develop in vivo gene correction therapeutics without unintended changes to the genome. When developing ARCUS, our genome editing pioneers looked to nature for a better editing system, better specificity, accuracy, and versatility. Our team started with a natural enzyme called ICRE1, which evolved in nature to specifically perform precise gene edits in a eukaryotic organism, algae. We delivered a process to engineer ICRE1 and reprogram the enzyme to target new genetic sites. This is what we call ARCUS. ARCUS has 4 key advantages. Safety: the enzyme has a natural off switch and doesn't edit DNA unless it is productively bound to its target site. Delivery: ARCUS is only 334 amino acids, and the small size permits delivery either by LNP or AAV. Control of edits: because of the unique type of edit, ARCUS can be applied to efficiently knock in or knock out target genes of interest. And it is proprietary. Precision has full control and freedom to operate. I'll now walk through a few examples of applications of ARCUS for in vivo gene editing and large animal models. These data demonstrate the durability of genetic edits made by ARCUS. Familial Hypercholesterolemia is a rare genetic disorder which leads to high level of serum LDL. And Precision has developed an ARCUS nuclease which knocks out the PCSK9 gene. Data on this slide demonstrate that one-time delivery of a PCSK9 ARCUS-encoded AAV resulted in 85% reduction in serum PCSK9 and a corresponding 56% reduction in LDL in non-human primates. This work was conducted in collaboration with Dr. Wilson's lab at the University of Pennsylvania, and the 3-year follow-up data was recently published in Molecular Therapy. At greater than 3 years out, we are seeing a stable gene edit without any obvious adverse effects. The edit is being inherited by subsequent generations of hepatocytes, and evidence supports that this is a permanent change to the genome. One of the reasons that our team has chosen to work with ARCUS is that it is very specific. ARCUS can discriminate between even a single base pair difference in the target DNA sequence, as demonstrated on this slide. ADRP is a disease caused by a single base pair mutation on one copy of the patient's rhodopsin gene and leads to degradation of the retina and eventually blindness. Data on this slide shows that the use of ARCUS in a transgenic pig model to knock out the PH23 mutant allele while leaving the wild-type gene intact results in restoration of rod function and vision within these animals. Primary hyperboxeleria type 1, PH1, is our lead in vivo gene editing program, and it is really characterized by a rare disease by accumulation of calcium oxalate in the kidneys, which leads to painful kidney stones and ultimately end-stage renal disease in these patients. A combined liver-kidney double transplant is often required to treat this condition. This slide shows data from a non-human primate study in which the ARCUS HAO1 nuclease was delivered through a one-time IV administration. Treatment reduced HAO1 by greater than 90%, GO protein levels by greater than 80%, and increased serum glycolate levels significantly, reducing downstream production of oxalate. This program has been prioritized by Precision and is one of our most advanced in the in vivo gene correction programs. We anticipate providing an update on the PH1 program in the first half of this year. Each in vivo editing program has a unique set of technical challenges. We've demonstrated efficient editing in vivo for knockout approaches in the liver and eye through published PCSK9 and ADRP work. And we are moving into applying ARCUS to diseases affecting other tissue types, including muscle. In addition to gene knockouts, ARCUS is particularly well suited for gene insertion and repair applications. The enzyme at the heart of the ARCUS platform evolved in nature to insert DNA within a large eukaryotic genome. We will continue to apply learnings as we overcome technical challenges, furthering the platform and unlocking additional programs. We are very excited about what we have already achieved at Precision and about what is still to come. Our company is made of a tremendous team, including global pioneers in gene editing, who have developed a truly differentiated gene editing platform, ARCUS. We anticipate achievement of a number of milestones in 2021 across our cell and gene therapy human therapeutic programs. Leveraging ARCUS, we are rapidly progressing the development of human therapeutics for serious diseases. We have already generated initial clinical data that validates our approach in allogeneic CAR-T, and are advancing a portfolio of in vivo gene correction programs addressing areas of significant unmet need. We are well funded and have cash into 2023, which will allow us to continue to push forward to realize our vision of improving life through genome editing. Thank you for your attention today. Given the time constraints, this presentation, including a subset of information found in our full corporate slide deck. Please visit the Precision BioSciences Investor Relations website for the full corporate slide deck, as well as the most up-to-date information on the company and pipeline.